So this is a talk by Jonathan Gropp and Itai Halevi from Weizmann Institute of Science. Metabolic isotopic model for biological methane production.
Thank you, Doron, for having me. Thank you, everybody, for coming. So I'm going to talk about my project with Itai Halevi in the Earth and Planetary Science Department here in the Weizmann. And I'll present to you a model about biogenic methane production, how we link physiology and geochemistry. So methanogenesis is the name for microbial production of methane, and this is the last step of organic matter degradation in the carbon cycle on Earth. So there are mainly three pathways of this process. It can be from CO2 reduction by hydrogen or from reducing acetate and methanol. I will focus today on CO2 reduction pathway, which is very common, and it has been conserved over probably about 4 billion years during the evolution, during microbial evolution here on Earth, and it is one of the most ancient metabolisms. Essentially, methane is just a byproduct of carbon fixation by these organisms through the Wood-Langdale pathway, and of course, energy conservation by creating a sodium ion gradient across the membrane. And one of the main tools that people use to study the sources of biogenic or abiotic methane is the stable isotope composition of methane. So we can use the ratios of the carbon to hydrogen stable isotopes of methane, which define specific fields of, for example, microbial CO2 reduction or microbial methyl type fermentation or abiotic sources of methane. Or we can also use the offset between the isotopic compositions of CO2 and methane, which also define distinct ranges of offsets for lab cultures or terrestrial samples, marine samples, etc. These are very useful, but these are also empiric, and they often overlap, these fields overlap, and they do not give us a unique solution for the source of methane. And we currently lack some understanding about the fundamental processes that lead to these isotopic signatures. And this was actually the goal of our project, to try to link between microbial physiology and these isotopic signatures. And the main candidate that appeared in recent years is that the thermodynamic drive of methanogenesis is causing these ranges of microbially produced methane. And the goal in our project was to develop this bioisotopic model of methanogenesis that will allow us to link between the thermodynamics of the process to the isotopic compositions, and will maybe allow us to study this process in environments that are inaccessible in lab cultures. I will just say that these kinds of models are essentially metabolic models that have a layer of an isotope mass balance, so they allow us to predict the isotopic compositions of methane for a given concentration of methane, CO2, etc. So the example question that I want to focus on today was that in some environments, there seems to be an apparent isotope equilibrium, specifically in energy-limited environments, such as very deep sediments in marine environments. And we wanted to know if we can explain these observations merely by slow methanogenesis, or we need some methane cycling or diffusion or mixing to explain these observations. So the model is what we do is we take this pathway. This is the representation of the metabolic pathway from CO2 to methane. Every step is an enzymatically catalyzed reaction, and we actually solve numerically this model to find the concentrations of all the metabolites for a given delta G. So the delta G is a function of concentrations of methane, CO2, and hydrogen, and we can then link the delta G of the reaction to the isotopic offset between CO2 and methane that I showed you before. So that's the same range of lab cultures that I showed you between 20 and 90 per mil. You can see that here the dependency of this offset is on the delta G. It's an inverse dependency where it is decreasing with the rate of methanogenesis. So these are all results from lab cultures, and we are very happy that the model in the blue line here captures the trend that is observed in lab cultures. And that's cool, that's very neat, but we were actually interested in what happens in terrestrial and mostly in marine environments that are severely energy limited and are actually inaccessible in lab cultures. You can see here that there is no data for very low delta G. That is, if we take hydrogen concentrations for too low levels, we cannot grow these microbes in the lab. So we wanted to see if our model actually can give us a good prediction what happens in these environments, and we started by calculating the cell-specific rates of methanogenesis. So we compiled some environmental data and lab culture data and found that the cell-specific rates, this is in units of femtomole per cell per day, span between 10 to the minus 5 femtomole per cell per day all the way to 10,000 femtomole per cell per day in lab cultures. That's a huge range, but you can see clearly that lab cultures are separated from most natural environments. So they do not necessarily reflect what's going on there. And we were happy to find that the prediction of the cell-specific rate from the model, that's the contour, as a function of temperature and concentration of hydrogen, is actually within the exact same range that is observed in marine environments, between 10 to the minus 5 and 10 femtomole per cell per day, which just for scale, 10 to the minus 5 femtomoles per cell per day is equivalent to 5,000 methane molecules per cell per day, not moles. So this is a tiny amount of net production of methane that is quite common in marine environments. And if we take it to energy utilization, we get this energy consumption of 10 to the minus 21 watts per cell. And this is more than 5 orders of magnitude lower than the lowest estimates of minimal threshold for maintenance energy in lab cultures. So we need to understand that lab cultures are fine, but they do not represent what happens in these severely energy limited environments where microbes actually thrive. And I'm going back to the question that I've started with, is that in many environments that are energy limited, for example, marine environments or shale gas and coal bed gas deposits, these are naturally accumulating gas deposits from microbial sources. They seem to fall on this 0-0 intersect of the isotopic offset between CO2 and methane, or methane and water, which means that there is apparent isotope equilibrium in the methane, water, CO2 system. And we wanted to know, again, whether it's possible that slow methanogenesis is producing these observations, which is not trivial, because biology is usually escaping equilibrium and it should not produce equilibrium signals. But we found that here, these are, again, the same range that is, I circled it here. So you can see the same samples over 10 to the minus 3 femtomole per cell per day is capable of producing equilibrium isotope in the CO2 water methane system, therefore proving that we don't require methane cycling or diffusion or mixing to get these signals. So what we found is that our bioisotopic model actually enabled us to simulate methanogenesis across more than 10 orders of magnitude of cell-specific respiration rates, which is really nice because we can tap into a range that is inaccessible in lab cultures, and we can actually tune some of the parameters such as cell size, energy availability, etc., to actually fit it to specific environments. And then again, deep sediments can actually produce methane that is at isotope equilibrium due to slow methanogenesis. And with that, I'd like to thank you